All right, seventh graders. So today we're going to start on the actual molecule of DNA. So we've talked about DNA for a lot this past year. So we just got off of doing traits and how traits are passed down from parent to offspring, right? In these traits, we talk about hair color, whether you have hair or you don't have hair, right? Skin color, all of these different traits that we've been talking about. And so now we're going to go kind of backwards and figure out what is making these traits. Like, what's the molecule that's making these traits? And that molecule is what we call DNA. And it's really at the central of all of biology, or life sciences, as we call it here in the seventh grade. Okay. So what you guys should be doing, if you haven't done it already, is I have emailed a copy of this DNA reading here. So you're going to go ahead and read this and make sure you check out what nucleotides are. Make sure you know what those are. They're composed of these three main parts, right? We have our phosphate group. Let me see here. Sorry, I got a whole bunch so you guys are going to be doing a cutout activity, and there's a bunch of cutouts. So I'm trying my best to make sure it's all squared away before I send it in. But we have these little phosphate groups, which are the plus signs, and these are the phosphate backbones. So they kind of hold all of the nucleotides together. Then we have something called the deoxyribose sugar. So those are these guys right here. That's why it's called DNA, for the deoxyribose part. And then right here it says we have the nitrogen-containing base. And so that's what really differentiates all of these nucleotides. And so every single nucleotide in your DNA has deoxyribose sugar and has that phosphate, has your phosphate backbone that holds all of them together. And then sprouting off of these two guys, we have our nitrogen-containing groups. So we have something called adenine, or you'll see this abbreviated as an A in your reading. Here's your thymine. So adenine and thymine, these guys will always match up together. And so here in this little cutout activity you guys have, you can see how one has a pointed end and the other one has a V, so they would fit together, right? So those are two nitrogen-containing bases. And then the other two are, we have guanine here. And so notice the shape. It has an outwards kind of lump on it. And then the other one is cytosine, which has the inwards. And so you guys are going to see all of these. Anyways, if I go ahead, and so like I said, by now you should have done all of this reading here. I'm kind of getting ahead of myself. So it starts talking about, here we go right here. It starts talking about adenine A, guanine G, thymine T, and cytosine C. Okay. And then it has this picture right here. Make sure you guys keep this and you read this part. So this will come in handy for mutations at the end of class. There's a couple questions on mutations in this paragraph here I typed up for you guys to help you out with that. Let's go ahead and jump over to our cutout activity. So here's those guys that I was just showing you. So here's our four nitrogen-containing bases, right? Adenine will always go with thymine. Guanine will always go with cytosine, and vice versa, right? If you see cytosine on one side of your DNA, then you'll see guanine on the other side. If you see thymine on one side of the DNA, then you'll see adenine. And what do I exactly mean by on the other side? Well, our DNA molecule is like a ladder. So if you can imagine, if you're crawling up a ladder, right, we have rungs that go across like this. And then we have the backbone of the ladders that hold those rungs in place, right? And so as I'm climbing up the ladder, I'm actually grabbing onto these nitrogen bases. I'm actually grabbing onto the adenine and the thymines that connect across. And I'm grabbing onto the guanines and the cytosines that connect across. And those are my rungs to my ladder. Okay, and so let me go back to my reading here. And so if we see this guy, here's a little more complex model of what a DNA molecule looks like. So this tannish gold part back here, here's our phosphate backbone, or the sides of the ladder that holds the rungs into place. And then these molecules in between here that we see, the little spheres and the little balls and stuff like that, those would be our adenine, thymine, guanine, cytosine. Up here, we can see that a little bit better if I can kind of zoom in. So we see the sides of the ladder, and then we see the rungs inside there. And like I said, that's the adenine, thymine, guanine, cytosine, and so on and so forth. Okay, and so what we're going to do with this activity is you guys are going to actually make your own molecule. So you can obviously see here that there's millions and millions and millions of these what we call base pairs. A base pair is this guy right here that I have for you, and the light's shining off it really bad right now. There it goes. So it has here's this phosphate backbone down here. Here's the deoxyribose, and then here's our first nitrogen base. And so this nitrogen base here sprouts off the edge here. Yeah. And then on the other side, this adenine will pair right to it because they fit together here. So you guys are going to create so one side that will have 4 of these guys. So you have thymine, guanine, cytosine, and adenine. So you'll have 4 of those guys down, and you'll create something that's starting to look like this. Okay, so I have my deoxyribose sugar here, and then attached to my deoxyribose sugar here, I have my adenine. And so here on this model, you see that we have a little dot and they have a little dot. And so that's how you know that this is where they connect, because a dot and a dot will connect. Then we see this one right here on the deoxyribose sugar is a square. Okay, so on my phosphate backbone here, I see another square right here. So I know that these 2 guys will connect. So I'll put those guys together. And so you guys can use glue. So there's those glue sticks on the back of my metal cabinet back there by my desk. You can use those stick glue, or you can use staples, or you can use scissors to do that, whatever is free to you. And then I'm going to go ahead so right here is one nucleotide, okay? A phosphate, deoxyribose sugar, and then the nitrogen base, which is adenine in this example. Next, I'm going to go ahead and keep on making my DNA strand longer. So my DNA strand is going to grow. And so now I see this star here, and this star will connect to this star. So I go ahead and connect those 2, okay? I'll take another nitrogen base like thymine right there. I'm going to connect this other guy right here, and it's repeating. That's all it's doing. Right here, and then finally right here. So star to star, circle to circle. Okay, and so like I said, in a DNA strand, we can have, this is a base pair right here. This is 1 base pair, this is 2, this is 3, this is 4. So we're showing 4 right here in your DNA molecule, in 1 DNA molecule, right? So 1 DNA molecule wraps up and forms 1 chromosome, right? If I show this to you, we have our DNA molecule right here, and this DNA keeps wrapping up, condensing, and it's coiling up until it forms this chromosome. So this is 1 DNA molecule here. How many of those do we have? We have 46 total, right? 23 pairs. And we've known this, we've talked about it for a long time. Okay? And so let me go ahead and go back to this. So now you're going to notice that we only have 1 side of this. So you guys are going to make this, and then you're also going to fill in the other side. So what pairs with adenine? Thymine, right? So and you're reading, you should have read about this. Adenine will always pair with thymine. So you'll have a thymine over here. And then down here you have thymine will always pair with adenine. Down here, guanine will pair with a cytosine. This cytosine will pair with another guanine. And so what you're doing is you're making we call it a complementary strand. So you're gonna cut out 1, 2, 3, 4 more deoxyribose sugars. You're going to cut out 1, 2, 3 more phosphate backbones, and you're going to cut out 1, 2, 3, 4 more nitrogen bases here, and you're going to create another DNA strand, or really half of the DNA strand, that's going to go over here, and you're going to connect these and make that ladder that you can climb up the rungs with. Okay, so it should look like a ladder, and it will be slightly offset. If you see it right here, it doesn't go exactly straight up and down. It's offset and it starts going at an angle, and that's because a DNA is a double helix, meaning as it goes, because of the structure of the molecule, it begins to twist. Okay, and that's you do need to know that, but we're not going to talk about why necessarily it does that. Okay, and so really that's your activity, is you're going to go ahead and cut out these guys, and you're going to glue them together. Once you guys are done with that, you can go ahead and get with people in your group. And then you can attach these. So you're going to have 4 base pairs, right? And so then you can attach it to your partners. They have 4. Now you're going to have a DNA molecule that's 8 base pairs long. Then you can go to another group, and they'll have 8 between them, those two. And then you'll have 16 base pairs on your DNA molecule. And then you can do your whole class together, right? Then you'll start getting this massive DNA strand that would then go in our bodies, right? And start coding for these different proteins that make the traits that we see. Okay. And so once you get done with that cutout activity, let me see here. Where am I at here? I got a bunch of stuff pulled up. Okay. So then you guys have your DNA cutout student activity PDF. So it has a simple procedure here, so you guys can have this while you're looking at it. So you're going to arrange the cutouts before lab. So make sure when you guys start doing all these cutouts like I had it there, make sure you have them laid out there nice before you start gluing and taping things together, or else if you do it wrong, then you're going to have to tear it all apart and maybe have to cut out new strands, and you guys don't want to do that. I almost forgot this part. You guys are going to need to color these right here. So adenines, all of your adenines that you put on there will be green. All of the thymines will be red. All of the guanines will be yellow. And all of the cytosines will be blue. Right now would be a great time to write that on the board. So adenines are green, thymines are red, guanines are yellow, cytosines are blue. So everybody remembers that, because I know somebody's going to be falling asleep right now, not listening to me. Okay. So next, here's the procedures. Then here's the questions. What does adenine pair with, right? That nitrogen group. What's guanine pair with? Describe the shape of the DNA molecule. Draw a picture if it helps. So you're going to go ahead and finish these questions here. Write a summary of the structure of the DNA that at least includes the terms base, sugar, phosphate. So the base is like the adenine, thymine, guanine, cytosine. The sugar is the deoxyribose sugar. The phosphates, the nucleotides. And then here it just says complete the other half. So what pairs with adenine? Thymine. What pairs with thymine? Adenine. So your DNA molecule will end up looking like this. But then again, like I said, you're going to create the other half of the DNA molecule so that then they are stitched together essentially, or you're going to tape them or glue them together. Probably tape them together or staple them together would work too. Okay. So once you go ahead and you get the student worksheet done, after you get done cutting everything out, you guys are going to go ahead and be moving on to mutations here. And so I have a mutation slip here. So make sure you guys pay attention, because you're going to have a DNA mutations exit slip that asks you about, please explain how mutations can affect the synthesis of protein. So when it talks about synthesis, when you synthesize something, you make something. So I specifically have a part in that reading about that question. Explain the relationship of genes, DNA sequence, and the shape of a protein. Okay, so like I said, I put all of that in your guys' reading. Why is DNA called the molecule of life? So here's all of these questions. So anyways, what you can do with this mutation slip here is you can staple this to your student handout for the cutout activity. You can hand this all in together. And this will be due Friday when you guys come back to class. But let's go ahead and start in on our mutations. So if you need to pause this right now and start doing your cutout activity, go ahead and do that. All right, but we're going to go ahead and get started on mutations. So we see right here, we see this mutation, this person's hand right here. So this can actually happen with your hands. It can happen with your toes. We call this web toes, web feet, right? This is actually fairly common. I remember I've seen this more than in one friend of mine, actually, so I'm not exactly sure how common it is. So there's really no webs. Like, you're not going to spread your fingers apart and there's going to be a thin skin there. That's not really how it works. They're just kind of, your fingers are stuck together, and that's web fingers or web toes. So anyways, we look at our chromosomes in our DNA. And like I said, in our bodies, in human beings, right, we have 23 pairs, or we have 46 total chromosomes in each of our cells, right? And these chromosomes are located within the nucleus of our cells. And so we see our chromosome here, and our chromosome is made up of DNA that's coiled up over and over and over, and these DNA molecules are long, right? So do you see a mutation? So we see here, we have our gene right here. We have DNA bases. So there's our adenine, thymine, guanine, and cytosines. And so what goes on here is this DNA will end up making its way out of the nucleus in a ribosome, right? If we really search back and think back to last semester, probably about October, we talked about all of the different organelles inside of the cell. And we talked about the ribosomes, where ribosomes are the site of protein synthesis. So ribosomes are the reason why proteins are made, and then proteins go on and make our traits, right? So we got to keep that in our head here. And so if DNA is changed, or we have mutation equals change in the organism's DNA. Sorry, I'm not going to restart my video. You can go ahead and pause this right now if you need to. Go ahead and get your notebooks out and write this down. Mutation equals change in the organism's DNA. Okay? So if we change the DNA, then that means we change the instructions. If you change the instructions, then obviously you're going to change what you build. Okay. If I have this big instruction thing, I'm making this castle of Legos, right? And I have this instruction pamphlet that tells me how to make this castle of Legos. But then something goes wrong and they accidentally send me a changed copy of that, and it actually tells me how to make the Death Star for Star Wars. Then I'm going to make an entirely different structure with those Legos because the instructions have changed, right? And that's what can happen in our DNA is if the DNA changes, and then that set of instructions there changes, thus causing the proteins to change. If the proteins change, then they will do something different. And again, I have that written down in your guys' reading that you guys already had. All right, so down here, the final product. So you kind of follow this from left to right. And so we have this, it says down here, altered protein, meaning it's a changed protein. All right, so we have this mutation here. We have a couple different kinds of mutations. So gene or chromosome. And so what this means is, what type of mutation is this? If I'm looking at my normal DNA strand, okay, but then look at this. I'm looking through it again. And so right here we have 2 yellows and 2 oranges. Up here we have a yellow and we have an orange, right? So it looks like we added 2 extra nucleotides in this sequence. So this is a mutation. So we added 2 on accident. And when I say we don't do that on purpose. Obviously, our molecules inside of our cells do that on accident, right? If they're making these DNA molecules that are millions and millions of base pairs long, there's going to be mutations. Okay, so every time that your DNA is actually replicated because you're making new cells in your body are dying, and then you have to make new cells to replace those recycled cells, we're going to get some DNA strands that aren't exactly the same, and that always happens. So is this an insertion, deletion, or substitution? Well, right now we added 2 different nucleotides, so I'd call that an insertion because we're inserting extra nucleotides. Okay. So what type of mutation is this? So we have thymine here, we have adenine here. So remember that thymine and adenine nucleotides always pair up. Cytosine, guanine nucleotides always add up. Okay, so it says right here, if I look at my normal DNA strand and then I come down to here where it says A changes to C, so this adenine nucleotide somehow changes to cytosine. Okay, and so is this in the gene or the chromosome? This is within a gene, right? And so is this an insertion? No, we didn't really insert anything. Did we delete anything? No, everything's still there. Or did we substitute something? Well, we actually substituted an adenine for a cytosine. And so now thymine and cytosine actually do not pair together, right? Because we know that guanine and cytosine only pair with each other, okay? And so because of that, we're going to get some kind of funky new protein out of this mutated DNA. Okay, so let's look at something called sickle cell anemia. So your blood carries oxygen to all of your muscles, to all of your organs, because all of our tissues in our body needs oxygen. And so your blood is in a specific shape, like a donut. And a donut is the good shape for carrying a molecule called oxygen that we all know and love. In sickle cell anemia, we actually have a molecule. The molecule inside of your red blood cells are distorted, and it looks like a crescent shape, like a moon, like when the moon's not all lit up, right? And so we have a normal strand so over here it says GTG-CAC, and so that's guanine, thymine, guanine, cytosine, adenine, cytosine. And so you'll see these things like valine, histidine, leucine, tyrosine, proline, glutamic acid. Glutamic acid. Okay. GAG, notice how guanine, adenine, guanine make this glutamic acid. Guanine, adenine, guanine make this glutamic acid. This is the process of them making the protein. Okay, and we're going to get to that more on Friday. So we're going to kind of skip over the protein making process for right now. We're just going to focus on what DNA is and the structure of DNA. Anyways, there was a mutation here. If we look at this DNA here, we have GAG for glutamic acid, but then the A gets switched out for a T. So now we have GTG. That actually codes for valine. Okay, so now instead of having this glutamic acid, we have this valine here. And so now we're going to get a new protein. We're going to get a protein that doesn't make the proper protein that carries oxygen in our red blood cells. Okay. And so here's a picture of, a normal red blood cell here. It kind of looks like a donut, but then we have this sickle cell blood here. And this is it's improperly formed, therefore it can't carry oxygen, and that's obviously not good. And so that's due to a mutation in the blood. And you can actually inherit this sickle cell anemia from your parents. So this is caused by a single amino acid change in the hemoglobin. So hemoglobin is the protein that carries the oxygen. And so an amino acid is many little Legos that make up a protein. And so we change one of those little Legos because of the DNA was changed. And now that changes the overall protein structure of the hemoglobin. So then we have another type. So we have something called deletion. So we already did insertion. That was the first one. We just got done with substitution, right? Substitution was sickle cell anemia, where adenine was swapped out was substituted for thymine, and then that got us sickle cell anemia. But now we're going to do something called deletion. And so what's going on here is we're missing this nucleotide, and so now it's gone. So this actually happens with things called cancer. And so as your body begins to replicate DNA, sometimes things are left out. So cancer occurs when mutation causes cells to grow in an uncontrolled way. And so we see this isn't due to obesity. So we talked about those rats last week or 2 weeks ago that had that agouti gene that was turned on because of epigenetics. They had the tan fur and they were also obese, while they had the twin that was the exact same, but that agouti gene that made them obese was turned off. Okay, so then we have famous mutants, right? There's no such thing as these mutants. At least we haven't found them yet. You're not going to roll into a vat of radioactive slew and then come out with really, really cool powers. Most of the time, your skin will rot off and you'll die within about a week if it gets too bad. This happened to the people in Chernobyl, which is a place in Russia where a nuclear meltdown happened. So not all mutations are actually harmful. Some mutations, or actually a lot of mutations, just happen, and they're not harmful or beneficial. They just happen, but then our proteins go on as planned. And so we see right here, we get a slightly different skin color on these grapes. We have an albino parrot instead of the yellow parrot. This is actually, we see this in nature. So this is due to a mutation. We see this lizard here that has some back fins like this and some crazy fins on the back of their head, right? Is this going to be bad for their health? No, not necessarily for these guys. Could this mutation be passed on to their offspring? Absolutely, it can. And this is how we get new species, right? If you guys think about squirrels, right? If you go to another part, we have fox squirrels all around us. They're like tan kind of fur that live in town, but then you can go somewhere else and then you have gray squirrels, right? Well, you're like, okay, that's just a little species of a squirrel. Well, this is how these species are made, is through these little itty bitty mutations that happen over long periods of time. And then all of a sudden now you only have this group of squirrel, that have a gray fur coat that mate with these gray coat squirrels, and then you have these squirrels, fox squirrels, that have like a tan brown fur color, and they only mate with the same ones. Calico cat here. So are there other mutations? Yeah. So we already talked about these guys. So the hairy ears, I forget what the name of the mutation is, but these guys with hairy ears, it's only carried here on the Y chromosome, so the father can pass it on to the son, and this will only happen in men, not in women. Why? Well, do women have Y chromosome? No, they don't, so they cannot get this hairy-eared mutation. Here we have Patua syndrome. I'm not super familiar with this one, but if we go ahead and look through all of these, we see, right, we have pairs of all of our chromosomes. So here's a pair of chromosome 1, pair of chromosome 2, pair, here's a pair, here's a pair, here's a pair. We keep going, and then we go down to here. 1, 2, 3. We have 3 sets of chromosomes here, which is not good. We should only have 2 sets of chromosomes. And so on the on the 13th pair here, we actually only have we have 3 copies of these guys, where then 2 copies, 2 copies, 2 copies, so on and so forth. So this is just an extra chromosome. This is the same thing that happens in like trisomy 21, which is also known as Down syndrome, except this is on the 13th chromosome, where trisomy 21 happens 3 copies of the 21st chromosome. We have Creux-Duchat syndrome, which is a deletion. And so we can see here on chromosome 4, so it's showing right here where this line is, and we can see that this chromosome right here is getting a little bit shorter. So we're deleting some DNA. Here on number 5, it's really evident between this guy and this guy. This is our pairs of chromosome here. This chromosome right here is getting a lot shorter because DNA is being deleted. Fragile X syndrome. I'm not exactly sure if this is an insertion or a deletion. But on this one, they kind of have an elongated face. They can have some learning disabilities. They can have flat-footedness and things like that. So those are just some example of real mutations that can happen within populations and things like that. So again, if you change the DNA, that is mutation. We change the DNA, then that changes the instructions for proteins that are made. If those proteins change, and then we change how they make other molecules in our body. Okay? So anyways, what you guys need to go ahead and do now is fill out that mutation exit slip that I was showing you before this, which is right here. And then once you finish that, you can go ahead and staple that and put that with your student handout, this, for the cutout. And you guys will be good to go, and this is all going to be due on Friday. If you guys need any more help, please let me know. Go ahead and message me on Teams. I'll be by my computer all the time. Bye.